Good morning, students. Yesterday we are discussing regarding the meiosis. So meiosis is nothing but a diploid cell always participates to form the 4 haploid cells, that is our gametes. So here whatever the chromosomes which are formed, they align as a pairs. So pairs in the alignment in the metaphase plate, the paternal chromosome and maternal chromosome, they form the pair, and these will, in the meiosis one, they form the 2 different dyads, because chromosomes, so it is a reduction division, so chromosomes will separate, pairs of chromosomes will separate in both the poles. So this is all yesterday we have discussed. So here, when in the meiosis 2, the chromatids will separate now. Previously in meiosis 1, chromosomes will separate, the pairs of chromosomes will separate. Now here chromatids will separate. So genetic content, whatever it is present in each of that is one DNA molecule. So here you see completely one DNA molecule, here one DNA molecule, in each chromosome they are present. So therefore here from each dyad the 2 gametes are formed. So completely. So therefore the chromatids, when they separate, this will form one particular cell and this will form one particular cell, whatever the number of chromosomes may be. So here it is haploid. Similarly, another dyad, if it participates, another 2 gametes are formed or haploid cells are formed. So totally 4 haploid cells are formed in the case of meiosis. So this is a broad idea regarding the meiosis, where meiosis 1 is a reductional division, because the chromosome number get reduced to half in both the dyads. Whereas in meiosis 2 is an equational division. In equational division, the chromatids will separate and equally both the cells are formed. So we call that as equational division. Okay. So this is what we see the meiosis, and whenever they are forming a pair, they undergo crossing over. Homologous chromosomes undergo crossing over. One paternal, one maternal, both are called homologous chromosomes. So these homologous chromosomes, they undergo crossing over to exchange the genetic material, and then again they separate by changing the genetic material. If they exchange, then you see some recombination takes place in each of the chromatid. So those only will undergo these changes. So we see recombination in so that is why characters will segregate in the meiosis. So this is an overall picture. Let us see in detail, point wise, what are the changes takes place in each and every step. So continue the sentences what we are discussing. Okay, right. If suppose it is not reduced to half, chromosome number doubled during fertilization, very important, chromosome number doubled. That is not the chromosome number, exactly speaking, the DNA content. The DNA content, exactly it is double, because we already know the number of chromosomes always remains same. So whatever the DNA content gets doubled. Okay, and when does you see the chromosome number doubled? In S phase. The chromosome number gets doubled during fertilization. When fertilization takes place, both the gametes will fuse. So content of chromosomes in one gamete and the other gamete, both will fuse to increase the number of chromosomes. So chromosome number doubled during fertilization. Chromosome number doubled during fertilization and is reduced to half by meiosis. And is reduced to half by meiosis. That means, as I already told, in meiosis 1, it gets reduced. We call it the reduction division during formation of gametes. During formation of gametes. During the formation of gametes, it is reduced to half. Okay, right. So, if it is not reduced to half, number would go on doubling with each generation. If not reduced to half, if the chromosome number, is not reduced to half, if it is not reduced to half, then what happens? Number would go on, chromosome number would go on increasing. From generations. Go on increasing or doubling, would go on doubling with each generation. Go on doubling. You can write it as doubling with each generation. Okay, right. So this is what it happens if there is no reduction in the number. Next, formation of haploid phase is by meiosis. Always the formation of haploid phase is by meiosis. Formation of haploid phase is by meiosis. Because the diploid cell undergo to the formation of haploid cells. So fertilization restores the diploid phase always. Fertilization. So once the haploids are formed, they don't be in a haploid. Again by fertilization, they convert into diploid phase. So fertilization restores the diploid phase. Fertilization restores the diploid phase. Okay, right. So, so this is something about the introduction regarding the meiosis. So the scientist who observed the reduction of chromosome number in animals is August Weismann. Just note down the information regarding the scientist inventions. August Weismann. August Weismann. In 1887, he observed the reduction of chromosome number in animals. He observed the reduction of chromosome number in animals. Whereas Strasburger later Strasburger later in 1888 observed in plants. So first in animals, then in the case of plants. Okay, right. Who coined meiosis? The term meiosis was coined by Farmer and Moore. Farmer and Moore in 1905 coined meiosis. Okay. The cell which undergoes meiosis is called meiocyte. Cell which undergoes meiosis is called meiocyte. Cell which undergoes meiosis is called meiocyte. Okay, right. So, what are the features of meiosis? That is very important. Let us see some features of meiosis. How the starting of the meiosis to ending of the meiosis it forms, just in the 4 steps, the features they have explained. So let us see those features. Features of meiosis. Okay, so here nucleus divides twice, chromosomes divides once. See the statement, nucleus divides twice, nucleus divides twice and chromosome divides once. So chromosome divides once and single cycle of DNA replication and single cycle of DNA replication. So this is what generally we see. So therefore, here when does nucleus divides twice? In meiosis 1, the nucleus will divide, and in meiosis 2, the nucleus will divide. So nucleus divides twice, but chromosome always divides in the reduction division. In reduction division, chromosomes will divide. Chromosome divides only once, and only one time the interphase will happen until the completion of meiosis. Therefore, we see that the number of the chromosome get, the number of the content of the DNA get reduced, replicated. So that happens in the interphase, S phase. So single cycle of DNA replication in the S phase until the completion of meiosis 1 and meiosis 2. So always nucleus divides twice. Nucleus divides twice, whereas in chromosome always divides once. Next, it starts after the parental chromosomes have replicated. Meiosis starts after the chromosomal number after the chromosomal, that is parental chromosomes, DNA content. Chromosomes are replicated to produce identical sister chromatids. DNA content have replicated. Here you can write parental chromosomal DNA content, because DNA is present inside the chromosomes. So DNA content, chromosomes DNA content to produce identical sister chromatids. Identical sister chromatids. So this is in S phase. Here it is in S phase. Next, it involves pairing of homologous chromosomes meiosis involves pairing of homologous chromosomes and exchange of genetic material between homologous chromosomes. And exchange of genetic material between homologous chromosomes. So meiosis involves pairing of homologous chromosomes because they pair, I told you. So during pairing only will takes place in the metaphase of meiosis-1, and exchange of genetic material, the genetic material content always between the homologous chromosomes they exchange. How does they exchange? Let us see. How does the chiasmata formation, how does the chiasmata, and how does they exchange between that? We see between the homologous chromosomes. Okay. So next, the 4 daughter haploid cells are formed. 4 daughter haploid cells. 4 daughter haploid cells are formed, each with how many? Half. That means each with half the number of chromosomes. Each with half the number of chromosomes. Each with half the number of chromosomes of parental cells. Half the number of chromosomes of parental cells and half of DNA content of parental cells. Here half of the number of chromosomes of parental cells and 1/4 of DNA content of parental cells. And 1/4 of DNA content of parental cells. DNA content of parental cells. What is the meaning of this? Means chromosome number will become half. That is in meiosis-1. In meiosis-1 I told you it becomes half. Whereas the DNA content first it is when in the parental cells, when it is participated in one chromosome, the pair, we see 2 DNA content. Okay, so that will become half, and another chromosome. That means the pair of homologous chromosomes, whatever they become, they pair, that will become 1/4. So each one contains 2 DNA molecules. So pair contains 4 DNA. They become 1/4. So 1/4 of the DNA content will be present finally in the gametes, haploid cells. So haploid cells are always N. So they forms the DNA content. So such kind of phenomena we see in the meiosis. So this is only just a features of meiosis. Still we have not completed the explanation in detail. So we'll see the process in detail, but these are the features. How does the meiosis? So you better understand this one we are explaining step by step. Okay. So stages, how does the stages will take place? Let us see. The stages of meiosis. Stages. This is meiosis 1 and meiosis 2. Reduction division. Reductional division, equational division. Reductional division, equational division. Meiosis 1 and meiosis 2. So equation meiosis 2 is an equational division or homotypic division. Okay, right. So this is equal to mitosis. You can write that also. This is equal to mitosis. For understanding purpose, this is equal to mitosis. So let us see what are the steps involved in the meiosis 1. In meiosis 1, what happens? So we are discussing in detail about the meiosis 1. So in meiosis 1, karyokinesis 1 and cytokinesis 1 takes place. Here karyokinesis 1, cytokinesis 1 takes place in the meiosis. So in karyokinesis we see Prophase-I, M phase, you already know. So Prophase-I, Metaphase-I, Anaphase-I, excuse me, and Telophase-I. These will take place in the karyokinesis. After that, cytokinesis will take place. Okay, so it is a complicated and a lengthy phase. Meiosis 1 is complicated and lengthy phase. So because why it is complicated, exchange of genetic material takes place, again they unwind, again they will separate, all these things are complicated. So we will see what is that complication. So chromosome number is reduced to half. Here chromosome number is reduced to half. That's what we said. Why? Why the chromosome number is reduced to half? That is why reduction division. So they go for pairing and separation. So who will pair? Same, paternal and maternal, whatever they pair are homologous chromosome. Paternal to one side, maternal to another pole. They will separate. That is why we call it as reduction division. So let us see the prophase-I. First we discuss karyokinesis. In karyokinesis, prophase-I followed by metaphase. In prophase-I, if you see, there are different stages. Okay, so keep the heading karyokinesis. In that we are discussing. So all these we have to discuss. So prophase-I, the first one is prophase-I. In karyokinesis, we are discussing about the prophase-I. So prophase-I is an important phase, complex, longer duration. Prophase-I, it is an important phase. It is an important phase, complex phase and long duration. And longer duration it takes place. Next, this is divided into, the prophase-I is divided into prophase-I is divided into leptotene, zygotene, pachytene, diplotene, diakinesis. Leptotene, zygotene, pachytene, diplotene, diakinesis. These are the 5 phases, 5 divisions. It is divided. Prophase-I is divided in all these particular stages. See, very important. Prophase-I is the lengthiest, and this is divided into 5 different phases. Not phases, 5 different steps. So first, again we have to discuss about leptotene, followed by zygotene, pachytene in detail. So if you this is only the important phase. Once it is completed, remaining steps are similar to the mitosis. How does the pachytene and all the metaphase, anaphase, telophase, all those will take place. So let us see in prophase-I what is leptotene. Okay, so keep the heading leptotene. Okay, right. So in prophase-I, leptotene. So here, what are the changes takes place? The size of the nucleus increases. Here, the size of the nucleus will increase in the leptotene. Chromosome is more distinct. With the double-stranded DNA, we can observe. Chromosome more distinct clearly with double-stranded DNA. DS stands for double-stranded DNA. Okay, Next, chromosomes appear as slender threads. Appear as slender threads. Chromosomes appear as slender threads bearing a series of granule-like structures. Bearing a series of granular threads. Chromosomes appear as slender threads bearing a series of chromosomes appear as slender threads bearing a series of granule-like structures. Series of granule-like structures. You can write it as a series of granule-like structures. Okay, called as chromomeres. They are called as chromomeres. Chromomeres. Next, irregular arranged. These are irregular arranged or polarized towards the centrioles. These are irregular and arranged or polarized towards the centrioles. Towards the centrioles. Arranged towards the centriole side they are arranged. Okay. Next, compactation of chromosomes occurs in the leptoteen. Compactation of chromosomes occurs during leptoteen. Compactation of the chromosomes occurs during the leptoteen. So these are the changes which takes place in the meiosis. Compactation means the thread-like chromosomes, chromatin material, they become shorter and thicker. So you can clearly identify in the form of chromosomes. Okay, right. Next, zygotine. So what do you see in the zygotine? What are the changes? Okay, in zygotine, what happens? Here, the chromosomes still become shorter and thicker. The chromosomes becomes more shorter and thicker. Chromosomes become more shorter and thicker. Homologous chromosome pair in lengthwise, which is called synapses. Homologous chromosomes pair in lengthwise, called synapses. So in zygotine, the homologous chromosomes, synapses will take place. This occurs due by the formation of synaptonomal complex. This occurs, which occurs by the formation of by the formation of synaptonomal, by the formation of complex structure called, by the formation of synaptonomal, by the formation of synaptonomal complex. So this synaptonomal complex is responsible for, very important, it's a protein complex. So this synaptonomal complex is responsible for pairing the homologous chromosomes. Pairing the homologous chromosomes. So that is what we call it as synapses. Very important term, synapses. Synapses always occurs in zygote. Remember. Okay? Right. So pairing. Who will pair homologous chromosomes means genetically and morphologically which are identical, similar. Next. It occurs by three different methods generally. The synaptonomal complex, the formation of the pairs can begin at the center and they move towards the poles. So in bidirectional or unidirectional or multidirectional, however they can pair up. So what are the main event takes place in the zygote means synapses. Synapses takes place in the pairing. Synapse is nothing but the pairing takes place in the zygote. Okay, right. Next, after synapses, pair of chromosomes, they call it as a bivalent. After synapses, pair of chromosomes, we call it as after synapses, pair of chromosomes, we call it as bivalent. Pair of chromosomes are called bivalent. Okay, so here nucleolus also increase in size. Here nucleolus also increase in size. So bivalence means, what is called bivalent? Two chromosomes are in pairs, we call it as bivalent. So that pairing is called synapses. Okay, next, centrioles move apart, initiating the spindle formation. Centrioles move apart, initiating the spindle formation. So centrioles will start forming the spindles. So this is what the zygote in the changes occur in the zygote. Okay. Next, packaging. In packaging, what are the changes? Till now, what happened? The chromosomes paired, homologous chromosomes paired, and then they form the bivalence. Now, next is packaging. What will take place? Packaging. So it's the longest stage. Packaging is the longest stage in the prophase-I. In prophase-I, this is the longest phase. Okay. So, and stable period, and it is the stable period. Okay. Next, here chromosomes become more shorter and thickened. Chromosomes become more shorter and thicken. More shortened and thickened, and clearly appears as tetrads. and clearly appears as tetrads. Tetrad means 4, no? So because as it is divided, the DNA content is divided, we can clearly see it as a tetrads. So bivalent consists of 4 chromatids. Tetrads means pairs. Bivalent consists of 4 chromatids. How many? 4. Bivalent consists of 4 chromatids. Next, recombination nodules appears between non-sister chromatids. Recombination nodules appear between non-sister chromatids. Nodules appears between non-sister chromatids. That means where crossing over takes place, the site where crossing over occurs, So crossing over occurs between the non-sister chromatids. So here we see the crossing over. Chromatids is the unit of crossing over. So now in this non-sister chromatids means what is sister and non-sister chromatids? Just note down. In the pair of chromosomes, that is one is paternal, one is maternal. Paternal and maternal chromosomes, chromatid of one chromosome, chromatid of another chromosome, these two are called as non-sister chromatids. These two are called non-sister. In one chromosome, the two chromatids are called sister chromatids. So these two are called sister chromatids. One chromatid of one chromosome and another chromatid of another chromosome, we call it as non-sister. That means they don't belong to the same chromosome, we call it as non-sister chromatids. So that is what generally we see it as the crossing over. So chromatid is a unit of crossing over. So this occurs by the enzyme recombinase. The recombination occurs by the enzyme recombinase. So this enzyme favors the formation of crossing over, recombination. Okay. So the point of exchange where chromatids break and fuse is called chiasmata. The point of exchange where chromatids, the point of exchange where chromatids break and fuse, is called chiasmata. Is called chiasmata. What is chiasmata? Wherever the exchange takes place. Suppose this is what the chromosome. Here exchange takes place. This point where the exchange is taking place is called chiasmata. Here only recombination nodule is seen. So crossing over results in the formation of chiasmata. So very important words. Chiasmata is seen here. Tetrads. Okay. So recombination nodules. Remember, recombination nodules, that is recombinase, followed by tetrad and chiasmata. These are the important things which takes place in the events which takes place in the packetine. Okay, right. So diplotin and diakinesis. Still we have diplotin and diakinesis, and in those two what happens, and followed by metaphase-I, anaphase and telophase. Okay, all these things in the next class will continue. Okay, right. So thank you.